penicillin or rarely unicellular, example yeast, Saccharomyces. They are saprophytic, decomposers, parasitic, or coprophilous, that is growing on the seed. Is branched and septate. In Ascomyces, mycelium is branched and septate. The asexual spores of chlamydia produced exogenously on the special mycelium called chlamydiophores. So the asexual spores of chlamydia produced exogenously on special mycelium called as chlamydiospores. Chlamydia or germination produce mycelium. Chlamydia on germination produces mycelium. Chlamydia are the asexual spores basically. The asexual spores are called as cospores. Sexual spores are called as cospores, which are produced endogenously in sac like asci, that is similar ascus. Asca are arranged in different types of floating bodies called ascocarps. Asca are arranged in different types of floating bodies called ascocarps. Some examples are aspergillus, claviceps, neurospora. Aspergillus, claviceps, Neurospora is used technical and genetic work. Many members like models, truffles are edible and are considered delicacies. Models and truffles. So now I will use Ascomyces. Basidium mycetes. Commonly known forms of Basidium mycetes are mushrooms, brachyfungi, or puffballs. Commonly known form of Basidium mycetes are mushrooms, brachyfungi, and puffballs. Mushrooms, brachyfungi, and puffballs. The green soil and long trees. So in Basidium mycetes, the sexual spores are generally uniform, but vegetative reproduction by fragmentation is common. So vegetative reproduction by fragmentation is common. In Basidium mycetes, vegetative reproduction is common. Then Ascomyces, the sexual spores are formed, which is known as chlamydia, and are produced exogenously on the mycelium, which is known as chlamydiospores. They germinate through chlamydium or germination produce mycelium. Okay, and then the sexual spores are ascospores. So they do possess both the kind, which are produced endogenously. Now this is produced endogenously, and then chlamydia is produced exogenously. Then ascospores are produced endogenously in sac like asci, and then similar asci. The asci are arranged in different types of floating bodies, and these are known as ascocarps. And the examples of Ascomyces are Aspergillus, claviceps, neurospora. Aspergillus, claviceps, neurospora. Molars and truffles cross the members, which is edible and is considered as delicacies. Okay, as we come to the Basidium mycetes, club fungi is the common name of Basidium mycetes, and here they are known as mushrooms, brachyfungi, and plants. They grow on soil, on trees, and living plant bodies inside the eggless and flower. The mycelium is branched and septate, same like Ascomyces. The sexual spores are generally uniform. The Basidium mycetes is no sexual spores. Non-form, but vegetative reproduction by transplantation is common. Vegetative reproduction by fragmentation is common. The organs are absent with plasmogeny is brought about by fusion of vegetative or somatic cells. Both the main reproductive organs are absent. The plasmogeny of different strains or genotype. Different strains or genotype and different variations to genotype. The resulting structure is dikaryotic, which is ultimately give rise to basidium. So on basis of this vegetative or by the fusion of vegetative or somatic cells, we will get the plasmogeny, and the plasmogeny is composed of different strains or genotype, and the resulting structure is the dikaryotic, which ultimately gives rise to basidium. And now karyochemy and meiosis takes place in basidium. The basidium which just is known as dikaryotic, karyochemy and meiosis do take place for the basidium, producing 4 basidiospores. Taking spores are exogenously produced on basidium. So these basidium spores are produced exogenously on basidium. That is, they are produced on the outer layer of basidium, or known as also basidia. The basidia are arranged in floating bodies called basidial carps. So here we call the floating bodies with consisting of the arrangement of basidia as basidial carps. Some common members are agaricus, common mushroom agaricus, then Ustelago, which is known as plant, and Pusinia, which is like grass. Some common members are agaricus. Of these fungi were discovered, they were moved into classes they were belong to. It is also possible that a sexual and vegetative state has been given one name, and these are the determinants, and the sexual stage and other are placed under another class. Later, when the linkage was established, the fungi were correctly identified and moved out of Ditromystis. Once perfect stages of members of Ditromystis were discovered, they were moved to Ascomystis and Basidiomystis. The Ditromystis reproduce only by oocyte. Ditromystis also possess chlamydia and reproduce through chlamydia. The mycelium is separate and branched. So far in all the three subdivisions, we are saying that the mycelium is separate and branched. Some members are sapraphites or parasites. Unlike the before one, here we can find sapraphites and parasites. Sapraphites meaning live on dead organic matter, and parasites meaning they live on the living host, which while a large number of them are decomposers of litter and help in mineral cycle. This is very important. Most of the Ditromystes are the decomposers of litter and help in mineral cycle. Some examples are Alternaria, Calyptotrichum, and Trichoderma. Alternaria, Calyptotrichum, and Trichoderma. Alternaria, Calyptotrichum, Trichoderma. Alternaria, Calyptotrichum, and Trichoderma are the examples of Ditromystes. Other examples of Ascomystes are Aspergillus, Claviceps, and Neurospora. Aspergillus, Ustelago, and Pusinia. Then for Ditromystes, we have Alternaria, Calyptotrichum, and Trichoderma. Alternaria, Calyptotrichum, and Trichoderma. Kingdom planting. Kingdom planting includes eukaryotic, chlorophyll, and heterotrophic, unlike the normal feature of plant. Bladderwort and Venus flytrap are examples of insectivorous plants, and Cuscata as a parasite. Cuscata as a parasite. The plant cells have an eukaryotic structure with prominent chloroplasts and cellulose, mainly made of cellulose. The cellulose is the content which makes up the cell wall of the plants. We study the eukaryotic cell structure in chapter 8. Planting includes algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. So what does the kingdom plantae include? Kingdom plantae has certain divisions, and that those are algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. Life cycle of plant has two distinct phases: the diploid sporophyte and the haploid cametophyte. Diploid sporophyte is like the spore formation, and haploid cametophyte is by the gametes formation or the fusion of gametes that alternate with each other, meaning this leads to alternation of genesis. So the diploid sporophyte and the haploid gametophyte that alternate with each other. The lengths of the haploid and diploid phases and whether these phases are free living or dependent on other vary among different groups in plants. This phenomenon is known as alternation of genesis or alternation of generation. We will study the further details in the chapter of kingdom planting. Kingdom animalia. The kingdom is characterized by heterotrophic eukaryotic organisms that are multicellular and their cells lack cell wall. They directly or indirectly depend on plants for food. They digest their food in an internal cavity, meaning eating both dead and living, or like plants and animals, by ingestion of food. and they follow a definite growth pattern and grow into adults that have a definite shape and size. Higher forms show elaborate sensory and neuromotor mechanisms. So higher forms in the human system are like the kingdom animalia show neurotransmittory and sensory elaborate sensory mechanism. Okay, neurotransmitter mechanism and elaborate sensory mechanisms. Higher forms show elaborate sensory and neurotransmitter mechanisms. Most of them are capable of locomotion. Also, locomotion is movement. The sexual reproduction is by copulation of male and female, followed by the embryo. These are briefly introduced here. All of us who have suffered the ill effect of common cold or flu know what effects viruses can have on us, even if we do not associate it with our condition. Viruses did not find a place in classification since they are not considered truly living. If we understand living as those organisms that have a cell structure, the viruses are monocellular organisms that are characterized by having an inert crystalline structure. Inside they have a crystalline structure. Outside the living cell, outside the living host, they have inert crystalline structure. Once they infect a cell, they take over the machinery of the host to replicate themselves. Once they infect, then they take over the machinery of the host and keep replicating themselves, killing the host. This replication causes the death of the host, basically. Could you call viruses living or non-living? It's a question mark. Viruses means venom or poisonous fluid. Dmitry Ivanovsky in 1892 recognized certain microbes as social organisms of the mosaic disease tobacco or tobacco mosaic disease. These were found to be similar than bacteria because they passed similar but smaller than bacteria because they passed Baring in 1898 demonstrated that the extract of the infected plants of tobacco would cause infection in healthy plants and named a new pathogenous virus and called the fluid as Contagium vivum fluidum. Contagiated. In fact, infectious living fluid. Now, W.M. Stanley in 1935 showed that viruses could be crystalline, and crystals consist largely of proteins. They are inert outside their space host cell. Viruses are obligate parasites, meaning without a living host they cannot survive. So that's why viruses are obligate parasites. In addition to proteins, viruses also contain genetic material. Not only proteins, not only that they contain proteins and they need a host to live, they do contain genetic material, and that's what makes them more dangerous and more harmful. In addition to protein, viruses also contain genetic material that could be either DNA or RNA. Both are not present in any virus. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. So a virus is basically a nucleoprotein. What nucleoprotein and its genetic material is very infectious or is infectious. In general, viruses that infect plants have single stranded RNA. Like what all viruses infect plants have single stranded that infect the bacteria. The bacteriophages or the bacterial viruses are the viruses which infect the bacteria itself. Usually consist of double-stranded DNA viruses. That's what the one which is mostly highly infectious consists of the double-stranded DNA. So the bacteriophages consist of double-stranded DNA viruses. The protein covering or the protein coat called capsid is made up of small subunits called as capsomes. So the protein which is present in the virus is basically capsid, and these capsids are made up of small subunits known as capsomes. These protects the nucleic acid. So the capsomes or capsomes protect the nucleic acid. Viruses cause disease like mumps, smallpox, hepatitis, and influenza. AIDS and illness is also caused by virus. In plant symptom can be mosaic formation, leaf rolling, curling, yellowing, and vein clearing, dwarfing, and stunned growth. Viruses is mainly diseases like it's very infectious also. Here are two figures. One is of tobacco mosaic virus and another one is of bacteriophage. The red one is RNA and this is capsid. This is here is the head. Then the collar neck is absent and bacteriophage. This is the sheath. Tall virus. Now we are going to go, except for virus, we have more subdivisions which are not included in the kingdom classification. Why? Because they do not possess a cell structure, and anything which does not possess a cell structure is not considered to be living. Now in viruses. In 1971, Theo Diner discovered a new infectious agent that was smaller than viruses. This is first of all smaller than viruses also, and caused potato spindle tuber disease. The virus which we discovered, which is actually smaller than that, it was a free-moving RNA. It was found to be a free RNA. It lacked a protein coat, and yeah, that is, it was an infectious agent, is smaller than viruses, and caused potato spindle tuber diseases. Potato spindle tuber diseases was caused by virus. Viroids caused potato spindle tuber diseases. It lacked the protein coat that is found in viruses, hence the name viroid. So it's called viroid because it lacks the protein which is basically found in viruses. The RNA of viroid was of low molecular weight. As RNA is the only genetic material in virus, it's first of all also low molecular weight. Prions. In modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded protein. Prions are more dangerous just because in modern medicine also it's found and it's a neurological disease. So neurology doesn't spread that much, but here it is. This prions is spreading abnormally folded protein. The agent was similar to size in virus. These agents were bovine spongiform encephalopathy. Bovine spongiform encephalopathy is a common disease which is caused by the prions and is commonly called mad cow disease in Latin. This really spoils the mechanism of the cow, and a cow is hardly able to come back to how it existed once it gets infected by BSE, which is bovine spongiform encephalopathy. Bovine spongiform encephalopathy. Bovine spongiform encephalopathy. What is it? CJD, which is CR Jacob disease. CJD stands for CR Jacob disease. Very good. Then, now lichens. Lichens are symbiotic associations. Lichens will be used in the next chapter, but here, lichens are symbiotic associations, that is, mutually useful associations. Lichens are symbiotic. Symbiotic means it's useful for the other person as well as for itself, so that they are mutually understand or mutually useful to each other. Between what? Algae and fungi. So like lichens is like a term used here between any symbiotic association. Like we have algae and fungi. Later we'll study about the slothware also, which has with animals. And the algae and slothware, algae and fungi. Yes. Algal component is known as phycobiota. So what is phycobiota? Algal component. How to remember? Additive photosynthesis only is performed by algae, and hence P stands for photosynthesis and P stands for phycobionts. And this is the name given for algae. And fungal component as mycobiont. autotrophic and heterotrophic. Algae is autotrophic, fungi is heterotrophic, respectively. Algae prepare food for fungi, and fungi provide shelter and absorb mineral nutrients and water for its partner. Algae cannot absorb these mineral nutrients and water, so fungi does that. As fungi cannot prepare the food, algae does that. This is what is known as lichens, meaning the symbiotic association or mutual association among two organisms. Okay, and now algae provide and here fungi provide shelter and absorb mineral nutrients and water for its partner. So close is the association that if one saw a lichen in nature, one would never imagine that they are two different organisms. So lichen is a combination of algae and fungi, one of lichen is so close like that you cannot differentiate that they are made up of two different organisms. Lichens are very good pollution indicators. They do not grow in polluted areas. This means that the lichens only grow in pollution-free area. So that indicates that to grow certain plants, we have to maintain the environment clean and pollution-free. This is one such plant which gives us the awareness of the area which should be pollution, because it cannot grow in pollution, and this cannot grow at all in polluted areas. It must grow in non-polluted areas. Hence, it's called as very good pollution indicators. Biological classification of plants and animals was first proposed by Aristotle. Aristotle was the one who even came up with the idea of giving in differentiation, like he classified into plant, herbs, and shrubs, or like trees, herbs, and shrubs. On the simple basis of morphological characters. In Linnaeus later classified four living organisms into two kingdoms. He classified into two kingdoms, Plantae and Animalia. Though he classified in kingdom Mammalia. Bacteria are cosmopolitan in distribution, meaning they found anywhere in the or all over the world. These organisms show the most extensive metabolic diversity. Who bacteria? Bacteria may be autotrophic or heterotrophic in their mode of nutrition. Kingdom Protista includes all single-celled eukaryotes such as Rhizophytes, Dinoflagellates, Euglenoids, slime molds, and protozoans. Who include this Kingdom Protista? Kingdom Monera was of bacteria, now Kingdom Protista includes all single-celled eukaryotes. Single-celled, but they are eukaryotes, okay? Like the Rhizophytes, Dinoflagellae, Euglenoids, slime mold, and protozoans. Members of Protista and now members of Kingdom Fungi show great diversity in the structures and habitat. Most fungi are saprophytic in their mode of nutrition. Fungi are known to be saprophytic, they live on dead matter. They show Basidiomystes and Diplomystes are the four classes. Most fungi are saprophytic in their mode of nutrition. They show sexual and sexual reproduction. Lycomystes, Ascomystes, Basidiomystes, and Diplomystes are the four classes under this kingdom. The Plantae includes all eukaryotic chlorophyll containing organisms. Algae, bryophytes, pteridophytes, gymnosperms, and angiosperms are included in this group. The life cycle of plants exhibit alternation of generations. This is very much important where diploid sporophytes and haploidophytes come in contact. Though they are independent of each other, in some cases they go in coordination to each other as they are two different structures. And this gives us the beauty of the alternation of generation or alternation of genesis. Alternation of generations. Same gametophytic and sporophytic generations. The heterotrophic eukaryotic or multicellular. The heterotrophic eukaryotic multicellular organisms lacking a cell wall. Main defect is lacking a cell wall are included in kingdom Animalia. The mode of nutrition of these animals is a holozoic. These animals here is the universal class. They reproduce mostly by the sexual mode. Some acellular organisms like viruses, virus, as well as lichens are not included in five kingdoms. And we know the reason why it's not included in the five kingdom classification also. because it lacks its own true cell wall and what can be defined as living. There's also like if a virus cannot survive without a host adopting the crystalline form, or is it by the help of those proteins, and even the genetic material from virus is like formed in virus. It's no smaller than mysteries of science or like biology. Okay. Plant kingdom. In the previous chapter, we looked at the broad classification of living organisms under the system proposed by Whitaker in 1969. These dates are important, like even in the previous chapter, we saw the three animals. He was the first guy who saw this tobacco mosaic virus, and it was 1892 when they did that. Then some people named venom, like venom virus, and now by further more research now, MW Beijing Beijing MW Beijing in 1998 in 1899 proposed the quote virus and the contagion fluidum. Contagion fluidum was proposed by him. After him came the W.M. Stanley in 1935. First 1919, first 1892, then 1898, then this guy came in 1935. So you can see a huge gap that in the middle people are not search much about why this is there. It's just gone into like extinct and again it came as a topic suddenly in 1935. Yeah, it shows that the virus is crystalline, could be crystalline, and crystal consists largely of proteins. The crystalline structure is what made everyone curious, like how is it keep crystalline and how is it like existing in a cell very wisely and reactively, but outside it's so inert. It's like it cannot recognize it's still living like that. And then we have another great T.O. Diner, our scientist in 1971, who recognized viroids, which is different from viruses. Which makes it easy for anyone to study about any particular kingdom or classification, rather division. In the division we get the classes. So yeah, where he suggested the 5 kingdom classification, which is Monera, Protista, Fungi, Animalia, and Plantae. That was his suggestion, which was considered and is still studied till date. In this chapter, we will deal in further classification with kingdom Plantae, popularly known as plant kingdom Plantae, and popularly it's known as plant kingdom has changed over time and will be changing. Fungi and members of the Monera, Protista having cell walls have now been excluded from Plantae. Before what, just having a cell wall was considered like okay, you're a plant. But now even other features are considered. So Monera and Protista got kicked out from the plant kingdom. They have their own kingdom. Yeah, plant early classification plays in the same way. Some of the cyanobacteria that also referred to as blue-green algae are not algae. Cyanobacteria, which we refer as blue-green algae, they are not algae anymore. In this chapter, we will describe algae, bryophytes, pteridophytes, gymnosperms, and angiosperms under a plant. These are the divisions which arise in the kingdom Plantae. It is algae, bryophytes, pteridophytes, gymnosperms, and angiosperms. First, before that, let us look at classification within angiosperms to understand some of the concerns that influence the classification of system. Here where we can relate the first chapter with taxonomy. It comes into the play. Okay. The earliest systems of classification characters such as habit, color, number, and shape of leaves, etc. They were based mainly on vegetative characters or the androecium structures given by lineage. We propose this like we can observe the androecium structure or the vegetative characters and separate them according to which class they belong by the characteristics which they will show. Such systems were artificial, and this kind of system was termed as artificial system. They separated the closely related species since they were based on a few characters. But this didn't stand out that way because they were differentiating based on a few things like physical characters which will obviously change through time. They didn't go into the depth characters yet. So artificial system did fail, but at a point existed. separated the closely related species maybe based on a few characteristics. Also, the artificial systems gave equal weightage to vegetative and sexual characters. Okay, they did give equal weightage to. It's not acceptable since we know that often the vegetative characters are more easily affected by environment. This is the main reason the artificial system by the Linus got rejected because you cannot expect it from vegetative characters. And against this, natural classification system developed. Now, providing a computation for artificial system, natural classification systems. What natural classification system arise? Developed, which were based on natural affinities among the organisms and consider not only external features but also internal features. This is the key point. Now, what started to consider the internal features before artificial systems not consider of internal features, only external and morphological features. Now, we have natural classification system under which we do classification system where they even study not only the external features of the plants but indeed the internal features in them also. So we can see the difference. At present, phylogenetic classification systems based on evolutionary relationships between the various organisms are accepted. This assumes that organisms belonging to same taxa have a common ancestor. But then they are like, let's go for the ancestor history family tree. We divide them based on their family tree. We now use information from many other sources to help resolve difficulties in classification. The classification is not one end and it's not dividing into various groups. It's always based on new component defined, you classify it into different sections because you have to study its features, all its important features, internal, external, upper, lower, anterior, posterior, everything. And now these become more important when there is no supporting fossil evidence. What they mean is, now once we collect the information which we require to classify anything under phylogenetic classification system, we do gather some of our research material, and this comes into use when we have difficulties in resolving the classification. Maybe now or further, then you have to resolve it again into further more fragments. We might find it hard. That time these we do. What does it do? It's based on all observable characteristics, and the computer analyzes the observable characteristics here. And it assigns set specific numbers and codes to all characters, and the data are then processed. So in the codes or digits is what consists of the data of the plant. In this way, each character is given equal importance, and at the same time hundreds of characteristics can be considered, and none of them are repeated. Like if one characteristic code is 101 for blue-green algae, let's say, and it will not at all be repeated. It's the same for aspergillus. That's the point of numerical taxonomy. What are the three taxonomies so far? First, we can consider the artificial system by Linnaeus, the natural classification system by George Bentham and Joseph Dalton Hooker. Then we do have phylogenetic classification system. After that, we came out phylogenetic, like the ancestral classification or ecological classification. I believe economical relationships or evolutionary relationships which humans had to their ancestors. Based on those only we classify them under or according to phylogenetic classification systems. Then we have the great numerical taxonomy, which make beer and chemotaxonomy. One is a number, its structure, its behavior, and chemotaxonomy. The uses of the basic chemical constituents. That uses the chemical constituents of the plant to resolve confusions and are all used by taxonomies these days. This is also one of the modern usage because it's used by taxonomies indeed. And cytotaxonomy helps more in detail when it's at like anything at its baby stage or like embryonic stage. It's quite easy to apply this taxonomy, cytotaxonomy. That's why many botanists rely on cytotaxonomy for their answers. And that was all a brief out about how the plant kingdom works and what all they wanted to do with the kingdom classification here. Now let's study the origin of the Viticulus by kingdom classifications and what all he considered, what all comes under his consideration, and what all we can study. Now, first and foremost, the main important thing is algae. Algae are chlorophyll bearing, simple thalloid, autotrophic and largely aquatic. Algae. This is like very important characteristics or introductory part of an algae. What is an algae? Algae are chlorophyll bearing. By that one, like a word chlorophyll, we can understand they are autotrophic. Algae are chlorophyll bearing, simple thalloid. That means algae itself possesses thalloid, not only one algae. Algae as a whole possesses thalloid. Autotrophic and largely aquatic. They are largely aquatic, but the case here is they are found both in fresh as well as marine organisms. They occur in a variety of other habitats: moist, stones, soil, wood. They have their own habitats according to which they are already adjusted, and they do follow that, like moist soil, wood, and rocks or soils. Some of them also occur in association with fungi like they are like the symbiotic association. Some the phycobiont, which is the algae, do exist with the fungi, and that it's known as lichen, and then with sloth-bearing animals. The form and size of algae is highly variable. It's not stable. From colonial forms like wallworks and the filamentous forms like Eulothrix and Spirogyra. Wallworks form, the structure is like wallworks form on the colonial form together. Colonial form of wallworks. Example is wallwork. And filamentous form like Eulothrix and Spirogyra. Eulothrix and Spirogyra are filamentous in nature. Okay, filamentous forms like wood bodies. Scallops form massive plant bodies. They help in the massiveness or like the increase in the plant. The algae reproduce by vegetative, sexual, and sexual motile and gives birth to new plants on germination are undergo sexual reproductions. Okay, and it takes place through the fusion of two gametes. These gametes can be flagellated and similar in size as in Eulothrix. In Eulothrix, we can see flagellated as well as flagellated but similar size in isogamous reproduction, where we deal with the fusion of the gametes or the egg cells. We study Eulothrix and non-flagellated similar in size as in Spirogyra. Reproduction. Fusion of two gametes similar or dissimilar in size as in species is Eudorinum, termed as angiosperms. We have anisogamous consists of Eudorinum. One of the great examples of anisogamous is Eudorinum. Fusion between one large and one non-motile static female gamete and a smaller motile male gamete is termed oogamous. So we do get three classifications, that is isogamous, anisogamous, and oogamous. Now here you can see the whole diagram of the structures. This is the wallworks, which consists of daughter colony and parent colony. The Eulothrix, this is, and this is laminiform, which comes under Lycomycete. This is Chlorophyll, Phycomycete, Laminaria. This is Dictyota, and this is Fucus. Then here we have optate algae. It is Rhodophyceae. This is a Porphyra and Polyciponia. Okay, now algae are used in many of variety ways. For radiation of Earth is carried out by algae through photosynthesis. This is very important because half of carbon fixation is carried out by this algae means in food because it has energy rich compounds in it, and this is the basic food for all aquatic animals. Many species of porphyra, certain lists of algae which is used as food by marine creatures, and in that list we have major three names here we have studied that included that 70 listed items or the 70 listed algae which is preferred or eaten by the marine inhabitants. Okay, of marine algae produce large of hydrocolloids. So as this is an overview, we are understanding about brown and red algae which produce large number of hydrocolloids. And what is this hydrocolloid basically? Water holding substance. Hydrocolloid is more like a sponge. It holds the water. So water holding substance is nothing but hydrocolloid. And what kind of hydrocolloid is present in brown algae? It's algin or algae. And what is it present in the red algae? It's keragine. Keragine and algin are the two main water or hydrocolloids present in the algae, okay, which are used commercially. These are commercially used. Agar, one of the commercial product obtained from Gelidium and Gracilidia. Gelidium and Gracilidia come into Rhodophyceae, and Rhodophyceae we can find the Gelidium and Gracilidia, and these two we get something known as agar which is used for agar, which is used in the industries for creating ice cream and jellies. Yeah, ice creams and jellies. Now the main very highlighted point we have here, and it is chlorella. Chlorella is like an algae which is unicellular, very rich in proteins. Chlorella is very much rich in proteins, and this chlorella is carried as a food for space travelers. So space travelers depend on chlorella for their food and also photosynthesis, as it's an algae which is very much rich in proteins and very much rich in producing oxygen also. What is that specific algae? That is chlorella. This is carried by space travelers and food supplement. Here they are not going to carry it like as an oxygen, they will carry it like food supplement. Okay, and the algae, now we have the classes which is there in algae and whatever major Chlorophyceae, Phaeophyceae and Rhodophyceae. Chlorophyceae, the members of Chlorophyceae commonly called green algae. The members of Chlorophyceae are commonly called as green algae. So we have color-based algae, and why they are called those also we should be aware. This also comes under questioning. Chlorophyceae are green algae. The plant body may be unicellular, colonial or filamentous. In Chlorophyceae, it might be unicellular, single-celled, colonial like volvulus, round structure, filamentous like Spirogyra. Spirogyra and Ostilago are filamentous. Wait, when will I come under filamentous? Okay, wait. Eulothrix and Spirogyra are filamentous in nature. Now we have the usual grass green. They are so grass green due to what? Due to the presence of chlorophyll A and B. Yesterday's question. Due to the presence of chlorophyll in shapes which algae can possess anywhere at any time. Of different species. Most of the members have one or more storage bodies called pyrannoids located in chloroplasts. And these pyrannoids are present in the besides starch also. Though starch is the food material which they are storing, they do possess other material except for starch, and that is protein. Oil droplets also. Some algae do store food in the form of oil droplets. Green algae usually have a rigid cell wall made of an inner of cellulose, outer layer of pectus. This makes the cell wall very rigid, like boundary, full boundary. Vegetative reproduction usually takes place by fragmentation or formation of different types of spores. Fragmentation we do here the sexual reproduction in you. Fragmentation or by the formation of different types of spores. Asexual reproduction is biflagellated zoospores produced in zoosporangia. So by zoospores, flagellatory movement, the process movement, flagellated zoospores present in produced by zoosporangia. The asexual reproduction shows considerable variation in type and formation of these cells, and may be isothermous and isogonous. This is the same thing condition as discussed before. Now we have some commonly four known algae, like green algae, which is a must and foremost to know. Chlamydomonas and Spirogyra. We have Chara, then Wordworts, Charamki, Wordworts, and Elatrix. All these five come under the class Chlorophyceae. And next we are going on to Phaeophyceae. The members of Phaeophyceae or brown algae are found primarily as marine habitats. They show great variation in size and form. They range from simple branched filamentous forms, ectocarps, to profuse branched forms as represented by kelps. Kelps represent the growth of the plant. So here kelps first. You get to know that they show great variation in size and form. They range from simple branched filamentous form. So that one of those forms is the ectocarpus. Ectocarpus is simple branched and filamentous form. Ectocarpus is simple branched and filamentous form. The profuse branched forms as represented by kelps, which may reach a height of 100 meters. So here possess chlorophyll A and C. Now A and B was for Chlorophyceae, and for Phaeophyceae we have chlorophyll A and C. It also possesses xanthophyll and carotenoids as mentioned, and here major thing is up to the amount of xanthophyll pigment, fucoxanthin present in there. So we have in the xanthophyll pigment fucoxanthin. Under xanthophyll pigment we have fucoxanthin also, which is present. It's an important pigment, and due to this also this whole process is occurring. Food is stored as complex carbohydrates. In Phaeophyceae, food is stored as complex carbohydrates, which may be in the form of laminarium or mannitol. What are the two forms which the stored food exist? That is laminarium or mannitol. The vegetative cells have a cellulose wall, usually covered on the outside of gelatinous clothing of algae. So the cellular wall is usually covered with a cellular arrangement or gelatinous coating called as algae. The protoplast contains, in addition to plastids, a certainly located vacuole, a centrally located vacuole and nucleus. More added up to the forwards, you know that the Chlorophyceae, the protoplast containing addition of plastids. In addition to plastids, a centrally located vacuole and nucleus. Okay. Brown algae is biflagellated zoospores. What come on? Vegetative reproduction. Everywhere vegetative reproduction is with fragmentation and algae. There is no other chance also. But the one which varies is the asexual one and the sexual one. The vegetative doesn't change. So here we get to know that the asexual reproduction zoospores that are pear-shaped and have two unequal laterally attached flagella. So now to get to know that this most brown algae is biflagellated zoospores that are pear-shaped and have two Two unequal laterally attached flagella. Unequally laterally attached flagella. Sexual reproduction may be isogamous, anisogamous, or oogamous. Basic divisions. Union of gametes may take place in water or within the oogamous species. So here the mutation or mutation. The union of gametes may take place in water or ectocarps. There is also ectocarps. Okay, ectocarps are simple branched filamentous form. I see ectocarps, dictyota, laminaria, sargassum, and fucus. These are the major common elements of Phaeophysae. And now Rhodophysae. Now members of Rhodophysae commonly called as red algae because of the predominance of the red pigment or phycoerythrin. And now why are these called as red algae? Again, the predominance of the red pigment is known as R phycoerythrin. R phycoerythrin. Dominance of R phycoerythrin in Rhodophysae is the reason why Rhodophysae are red in color. One person has red algae. Majority of the red algae are marine. They are marine, but more of them are found in warmer areas. They occur in both well-lighted regions close to the surface of water and also at great depths in ocean where relatively little light penetrates or no light penetrates. With these two live. That's their specialty. The red thalli of most of the red algae are multicellular. The red color thalli of most of the red algae are multicellular. Some of them have complex body organization. Oh, Rhodophysae have complex body organization. All the three cases food is stored in different way. In first is stored as iron ore, second laminarian or mannitol, and over here it's Floridian starch, which is very similar to amylopectin and glycogen in structure. So we can say it has its complex body organization. It's quietly near to the human storage unit. So like humans store glycogen, and their reserve food is stored as glycogen. And when this reserve food is stored as glycogen, similar structure of glycogen, not exactly glycogen, amylopectin and glycogen. In structure, the red algae usually reproduce vegetatively by fragmentation. See, all the cases, fragmentation. They reproduce asexually by non-motile spores and sexually also by non-motile gametes. None of these have movement. Non-motile is which lacks movement, is static. You have post-fertilization developments. The common members of this class are Polyphyra or Polyphyra. Then we have Gracilaria, Angelidium, Geladium, and we have Polysphonium. Rhodophysae, and with that we are done with algae. Now we have bryophytes. Bryophytes is a huge interesting one, as bryophytes include the various mosses and liverworts. What do they include? Mosses and liverworts that are found commonly growing in moist shaded areas in the hills. They are seen in the moist shaded areas in the hill stations. Hills. So we have two key structures to study as liverworts is the questions. First we have the archegonium. Archegonium itself says the female antheridiophore, Gimacup and rhizoids. What's the difference? Only the antheridiophore and the archegoniophore. Archegoniophore and the antheridiophore is the main difference between the female and male structures. Archegoniophore is for female, antheridiophore is for male. Archegoniophore for female, antheridiophore for male. Right. Now bryophytes include the various mosses and liverworts found in commonly in moist shaded areas in the hills. Bryophytes are called amphibians of the plant kingdom. In animal we have frog, in plant we have bryophytes. Because these plants are important for sexual reproduction. Okay. Plant succession on bare rocks and soil. The plant body of bryophytes is more differentiated than that of algae. Compared with algae, thallus-like and prostrate or erect, and attached to the substratum by unicellular or multicellular rhizoids. So now as we study the shape, the parts, we can see what they're actually useful for. It has a thallus-like prostrate or erect and attached to the substratum down by unicellular or multicellular rhizoids. They lack true roots. So bryophytes have no true roots. It's different. So like when we go to the root system, we can read the true roots and the other root system. They lack true roots, stem or leaves. All these are like absent in bryophytes. They may possess root-like, leaf-like, stem-like structure. They may possess root-like, leaf-like, stem-like structures. The main plant body of the bryophyte is haploid. Main plant body of the bryophytes is haploid. It produces gametes. Main main plant produces gametes, hence is called gametophyte. Produces gametes, gametophyte. The 6 organs in bryophytes are multicellular. Here they are multicellular. The main 6 organ is the antheridium. Antheridium is for male, and antherizoids, they are also called this. With the they produce biflagellate antherizoids. They produce the biflagellated antherizoids. The female ones are called as archaeonium, which is flask shaped, and released into water where they come in contact with the archegonium. An antherizoid fuses with the egg to produce the zygote. Here we can see for the first time the zygote formation. Zygotes do not undergo reduction division immediately. So as soon as a zygote is formed, they don't go under reduction division. They are not going to die. They produce a multicellular body called sporophyte. As soon as the name is, they produce a multicellular body, and that body is known as sporophyte. The sporophyte is not free living but attached to the photosynthetic gametophyte and derives nourishment from it. As the sporophyte is not free living, it is attached to the photosynthetic gametophyte and derives nourishment from it. Photosynthetic gametophyte and derives nourishment from it. Some cells of the sporophyte undergo reduction divisions. Here the reduction divisions start. After all this of sporophyte, we can see some cells of sporophyte undergo meiosis, that is the reduction division, to produce haploid spores. Once again, to continue the cycle, these spores germinate to produce gametophyte, and again we can see the cycle continues. Haploid spores they are, and these spores germinate to produce gametophyte. Now we have bryophytes in general are of little economic importance, but some mosses provide food for herbaceous mammals, meaning herbivorous mammals, birds and other animals. Species of sphagnum. Sphagnum is very important species over here. It's a moss. Sphagnum is a moss which provide shipment of living material because of their capacity to hold water. Sphagnum. Mosses along with lichens are the first organisms to colonize rocks. Mosses along with lichens are the first organisms to colonize the rock, and hence are of great ecological importance. They decompose rocks, making the substrate suitable for the growth of higher plants. So they decompose the rock itself, making the substrate or like the land suitable for growth of higher plants. Since mosses form dense mats on the soil, they reduce the impact of falling rain and prevent soil erosion. Due to the mat formation of the mosses, this prevents the impact of falling rain and hence prevents soil erosion too. The bryophytes are divided into liverworts and mosses. The bryophytes are divided into two main sections like liverworts and mosses, and now we'll see what liverworts. Liverworts. The liverworts grow usually in moist, shady habitats such as banks of streams, marshy ground, damp soil, bark of trees, and deep in the woods. What do they grow in? Banks of the streams, marshy ground, banks of the streams, marshy ground, damp soil, back of trees, damp soil, so back of trees, and deep in the woods. The plant body of a liverwort is thalloid. Here also thallus came into the play. Because there are rhodophyceae here, liverworts. The plant body of liverwort itself is a thalloid. Example, Markantia. The thallus is dorsiventral and closely pressed to the substrate. It's dorsiventral and closely pressed, meaning closely in contact with the surface, the substrate. The leafy members have tiny leaf-like appendages in 2 rows on the stem-like structures. Asexual reproduction in liverworts takes place by fragmentation of thalli. Here also fragmentation, but it's of thalli. Nothing but thallus, or by the formation of special structures called gemmae, single gemma or gemma, gemmae, the thing over here, which is formation of specialized structures. This is only in liverworts, nowhere else. Gemmae are green. They are green. They are multicellular. Gemmae are green color, multicellular, asexual buds, which develop in small receptacles called gemma cups. Okay, they are green, multicellular, asexual buds, which develop in small receptacles called gemma cups located on the thalli. The gemma become detached from the parent body and germinate to form new individuals. During the sexual reproduction, male and female organs are produced either on the same or on different thalli. Like they undergo the sexual reproduction and it's either produced on the same or different thalli. The sporophyte is differentiated into. What is differentiated here? The sporophyte. What is sporophyte? It grows by produce a multicellular body after the zygote formation. This happens and the zygotes don't undergo reduction division, but they immediately do this. So the sporoid is differentiated into foot, seta, and capsule. Or seta we can call. Foot, seta, and capsule is what is differentiated into. After meiosis, spores are produced within the capsule. After meiosis, the spores are produced within the capsule. Okay, the sporophyte has 3 parts, which is foot, seta, and capsule. After meiosis, the spores are produced within the capsule itself. These spores germinate to form free-living gematophytes, and again the process continues.
ಬಟ್ ಇದು ರೀಚ್ ಆಗಲ್ಲ ಪ್ರಿಸ್ಮ್